Sure. So I was presenting a phase 1 first-in-human study with a novel bispecific antibody targeting FcRH5 in relapsed refractory multiple myeloma. FcRH5 is really a new target in multiple myeloma. It's a cell surface receptor that's expressed only in the B cell lineage, namely B cells and plasma cells. But the expression is highest on plasma cells, especially malignant plasma cells, as in myeloma. And it's almost universally expressed in myeloma patients, which makes it an attractive target for myeloma immune therapy. So the drug that was studied in this phase 1 study is called cevostamab, and it's a bispecific antibody. So one end of the molecule targets FcRH5 on the myeloma cells, the other end targets CD3, which is on T cells, and essentially this leads to activation of the endogenous T cells within the bone marrow microenvironment, leading to T cell mediated killing of the myeloma cells. And so based on promising preclinical activity in myeloma models, this phase 1 study was set up, and we're presenting the first data. This was a initial sort of typical 3+3 dose escalation design. And what was interesting about this study is that it entailed what's called a step-up dose escalation. And so one of the risks of bispecific antibodies is cytokine release syndrome. And so to try to mitigate that risk, this study first gave a low dose of the antibody on cycle 1, day 1. And then if patients were tolerating this, they'd get the full target dose starting on day 8. And then the drug was given once every 3 weeks as an IV infusion, starting with cycle 2, thereafter up to 17 cycles. And so initially the step-up dose was dose escalated from a very low dose, 0.05 milligrams, up to 3.6 milligrams. And then once the safety of that step-up dose was established, it was fixed at 3.6, and then we further escalated the target dose. And dose escalation is actually ongoing, and we're up to 160 milligrams and climbing. And as mentioned, the drug's given once every 3 weeks as an IV infusion. Patients are hospitalized for the first 2 doses for up to 72 hours to monitor for cytokine release syndrome and other toxicities. And then if tolerating well, they get the rest of the doses as an outpatient. They are pre-medicated with Tylenol and Benadryl and steroids with the first couple of doses to mitigate a infusion-related reaction. First, I guess I'll mention that the patient population included in the study was sort of highly relapsed refractory myeloma. It was a median of 6 prior lines of therapy in these patients. All of them had prior proteasome inhibitors and IMiDs. About 80% had prior CD38 antibody. 72% were triple-class refractory, and 45% were penta-drug refractory, meaning they were refractory to 2 proteasome inhibitors, 2 IMiDs, and a CD38 antibody. And interestingly, 21% of the patients so far had had a anti-BCMA therapy in the past, either BCMA CAR T cells or an antibody-drug conjugate. So the data that we presented included 53 patients who were evaluable to date in the single step-up cohort. And the primary findings of the study were that the main toxicities were cytopenias, which were seen in grade 3, 4, and 15 to 25% of patients. Cytokine release syndrome was seen in 76% of patients, although all of these but one were grade 1 and grade 2, which suggests that this step-up dosing was effective at mitigating the high-grade CRS. And neurologic toxicity was seen in 28% of patients. All of this was grade 1 or grade 2 and would occur in the setting of CRS and then resolve typically within a day or 2. Tocilizumab was given in 23% of patients, steroids in 17% of patients. And there was very little CRS after those initial day 1 and day 8 doses, and it was fairly well tolerated thereafter. In terms of efficacy, there were no responses in patients getting the lower doses of the drug below 10.8 milligrams or below, but starting at 20 milligrams as the target dose and moving up, which we consider the active doses, there were 34 patients who received these active doses, and 18 of them, or 53%, actually had a partial response or better, including 32% with VGPR or better, and I believe 18% CRs. And this included patients who were penta-refractory, including patients who had prior BCMA therapy, and maybe a hint of sort of greater activity as we got up to the highest dose levels, 90 to 132 milligrams. 61% of patients at those doses responded. So those were the primary, I guess, efficacy findings. The durability of response, I think, is still a little too soon to tell. The median follow-up, especially at the highest dose levels, is short, but we have multiple patients out beyond 6 months, several beyond a year, with ongoing durable remissions. And I think we just have to wait and see overall what the durability of response is going to be, particularly at some of the higher, more active doses. We're continuing to dose escalate in the single step-up dosing cohort. There's also been a small expansion at the 90 milligram dose. And so hopefully we'll have some additional data to present later this year. In addition, the study's now opened a double step-up cohort where they get an even lower dose on day 1, a medium dose day 8, and then the full dose on day 15. Again, trying to see if we can reduce the incidence of cytokine release syndrome. And so that double step-up dose escalation is ongoing. So ultimately, once the recommended phase 2 dose is found and we see which of these two approaches, single or double step-up, really looks the most promising based on the balance of toxicity and efficacy, then expected there'll be a large sort of phase 2 expansion to really get a sense of how effective the drug is going to be and better sense of safety, tolerability, and durability of response. Sure. So I think the main takeaways so far are, one, yes, it is early still, so I think we need more time and more data, but this does establish proof of principle that FcRH5 is a viable target in multiple myeloma. This is really the first study to show that you can get responses, including deep responses in highly refractory patients targeting this. So I think it validates the target. It also shows the activity of this novel bispecific antibody. But I think it's a little too soon to tell sort of where this drug is going to fit in our armamentarium of multiple myeloma. Obviously, we hope the phase 2 expansions validate and confirm the data that we're seeing in this early dose escalation period. And then once we know a little bit more about the response rates and the populations that are more likely to respond, that'll help guide us to where we're going to be using this going forward. No, I don't think so. I'd just like to really thank all of my co-investigators and all the patients who've enrolled in the study.